Hello, my name is Elizabeth Potrykus. I'm a freshman at Iowa State University studying industrial engineering. I'm working as an undergraduate research assistant for Dr. Lizhi Wang in the Industrial Manufacturing Systems Engineering Department. Today we just have a quick video for you to introduce plant breeding and all of its advancements to the world of engineering. So the main goal of a plant breeder is to cross together two different plants to produce an even better offspring. Past methods have worked in the past, but we need to optimize those to keep up with our ever-changing environment, and we're doing that by using engineering methods. So here we have a quick example. I have two peppers here, and by crossing them together, we're able to produce an offspring that is more red than that first one, and then larger than the second. And we hope by making the correct decisions, we're able to produce peppers that are even larger than the last. We know that crossbreeding has been working really well for us because ever since it began, you can see that the US average corn yield has been extremely increasing. So because we're going to be using corn for the rest of our examples, here's just a little bit of a background on how corn reproduces. Each plant has both a tassel and an ear with silk, so when pollen from the tassel goes to the ear with silk, it's able to produce that next generation. Now because it has both male and female parts, it's able to do what is known as self-pollination. Otherwise, it also has cross-pollination too if you want to use two different plants. So the first methods of crossbreeding was known as phenotypic selection. This is when we made decisions based off of the plant's phenotypes. Phenotypes being how tall it is, the color, its yield, things like that. So as a crossbreeder, we would have a population of corn plants, and then our job was to make the decision of which ones to cross together to produce our tallest next generation. So as phenotypic selection goes, we would pick the two tallest to hopefully produce the tallest generation. Now, although this method did work some of the time, it wasn't as consistent as we want it to be. As you can see with that produced generation, it wasn't always as tall as we predicted. So that's what led to conventional genomic selection. This is when we made decisions more based on the plant's chromosomes and their DNA. So as you can see with each seed, we're able to look at the chromosomes, and from those chromosomes, we're able to predict those plants' phenotypes. For example, how tall it's going to be when it's fully matured. Now we were able to make these decisions while they're seeds, and we don't have to wait around for them all to be fully developed to make that decision on which ones to breed together. So this saved a lot of time and money. Now this next picture just displays how complex genomic selection really is. Just in this first row, you can see how many plants there are and how many decisions we need to make, which ones to breed together, how many we should breed together. You can also see in this picture when they use cross-pollination versus self-pollination over here. So now just a quick biology background on those chromosomes that we're looking at to make those predictions. Here we have two sets of chromosomes, and we're going to cross them together to produce that offspring, also known as a progeny over here. Notice how half of them is more of that red line coming from the first parent, and then the other half is more of a blue color coming from the second. This is known as Mendel's first law. It states that 50% of a plant's chromosomes comes from one parent and 50% from the other. Now, since we know that only 50% comes from one of the parents, we're going to look at one parent and that 50% that we're given from that one parent. You can kind of see a copying and pasting effect here, how it copies from one line and pastes that right into the next, into its offspring. This is known as genetic linkage. However, occasionally it does jump to the other side of that chromosome, and then it goes on and continues that copying and pasting effect. That's what's known as genetic recombination. So now we just have a quick example going through that. We're going to cross together two plants here. This first one is known as an elite line. We're going to call it the elite line because it has the phenotypes that we want to get passed on. So we're going to represent the traits that we do want to get passed on and are really present in this plant with the yellow boxes. So this plant would have really good height and yield. However, the purple boxes are going to represent things that we don't want getting passed on to that next generation. So for example, this one would have the color that we don't want getting passed on. So we're crossing that with the second plant, and we're going to call this the donor line because it has mostly purple boxes, but those yellow boxes were in the areas that the elite line didn't have yellow boxes. So this one does have very good color, and we want that to get passed on. Now, cross them together and we get a whole new generation. This displays really well Mendel's first law, how 50% comes from either parent. So now what we're going to do is we're going to self-pollinate one of these offspring, and we're going to produce an entire new generation. So here's that being displayed. This would be a good time to pause the video and see where genetic recombination and genetic linkage is taking place. Now the next topic that I'm going to introduce is known as genomic estimated breeding value. This just assigns a score to each plant so we can easily make those decisions on which ones to breed together. So we're going to assign 2 points to the yellow boxes because those are phenotypes we want to get passed on, and 0 for the purple because we don't want those to get passed on. So we go through and calculate the GEB value for each of these, and the higher that value, the more likely we're going to breed them together. Now, plant crossbreeders don't make decisions for just a few plants. They have to make decisions for an entire population, so that's what we have here, an entire population of corn plants. And what we're going to do is we're going to assign a GEB value for each of these plants. Now these green bars represent that value, and the higher the bar, the higher the GEBV. So with genomic selection, we would want to be using some plants that are more towards this side. Just a quick summary of what we all went through. The first thing is just how important every single decision is when choosing which plants to breed together. We started off with phenotypic selection and then went into more complicated genomic selection, and now we're using even more advanced methods, and we do that through engineering methods. So engineers should really get involved. The next thing that I would like to mention is a competition that was introduced in one of Dr. Lizhi Wang's graduate level classes. He and a few colleagues wrote a paper with his findings, so I'd highly recommend that you go check that out. And that's all I have for you today. Thank you so much for watching. Dr. Lizhi Wang, Iowa State University, and all of our partial sponsors.